This is our first video lecture for Chapter 14, Chemical Equilibrium, and I'd like to start off with a demonstration of a chemical equilibrium. So here we're going to be looking at an equilibrium in aqueous cobalt 2 chloride solution and how it can be affected by heating. So it starts off by showing us the cobalt chloride, and they make an aqueous solution of the cobalt chloride. So the U-tube is being filled with the aqueous cobalt 2 chloride, and the chemical formula for this is this cobalt right here. So you've got a cobalt with 6 waters bound to the cobalt, and it has a 2+ charge. So the waters are solvating, or actually they're bound to the cobalt. Now if they heat the system, it's going to shift the equilibrium to the right, and it's going to form some cobalt tetrachloride species. So this cobalt tetrachloride species is a tetrahedral complex, and it has a blue color to it. Now in this situation where we've got the blue color, you have the cobalt tetrachloride, that's the predominant species in here, but there's also some of the pink cobalt hexaquose species. But on the other side, where we have the cobalt hexaquose species, is the predominant species in solution. There is also some cobalt tetrachloride in there as well. It's just that there's so little of it, we predominantly see the color associated with the cobalt hexaquose species. So the pink color that we see here, this is due to the cobalt hexaquose species. So that means that there's a cobalt complex, and this is an octahedral complex, and there are 6 waters bound to the cobalt. That's supposed to be a 2. And this complex has an overall charge of 2+. And this is what we call a coordination compound, and we'll discuss this a little bit more when we talk about Chapter 24 with the transition metal complexes. Now this hexaquose species that has the 6 waters around it, if I were to heat this compound up, I can shift the equilibrium. Because in reality, in solution, there's a mixture of this cobalt hexaquose species, this octahedral complex that's pink, and this cobalt tetrachlorose species. So there's a cobalt, and it's a tetrahedral complex. It's got 4 chlorides bound to the cobalt. And this complex has a 2- charge. So both the cobalts are in the 2+ oxidation state. But here each chloride is a -1, so there's 4 chlorides. So the overall charge, when I add up the 2+, the 1-, 1-, 1-, 1-, we get a 2- charge overall. So there's an equilibrium in solution, but going between these two species. So it's going back and forth. Now, at room temperature, the equilibrium lies closer to the left, and we see more of the cobalt hexaquose species. That's why it's pink in color, because this complex is pink in color. But if we heat the complex, if we heat the solution, it's going to drive the equilibrium to the right. And when it does, it makes more of the cobalt tetrachloride complex. And this complex has a blue color. So this is why heating it changes the color, because we've shifted the position of the equilibrium. But there's still an equilibrium happening where you get the conversion between these two different species. And this up here is just the balanced chemical equation showing the chemical equilibrium. So to explain where this color comes from, one of the things that we'll learn later on is that the d orbitals of the cobalt complex are actually split. So there are 5 d orbitals, and because it's a cobalt 2+, there are 7 d electrons. 1, 2, 3, 4, 5, 6, 7. So this is how the orbitals are split in an octahedral complex. But in the tetrahedral complex, the order is different. So there's 3 on top, 2 on the bottom. There's a difference between these energy gaps. The one for the octahedral is larger than for the tetrahedral. So I'm going to go ahead and draw all the electrons for the cobalt, because it too is a cobalt 2 plus, so that's a D7, 1, 2, 3, 4, 5, 6, 7. So if you'll remember from lab when we're talking about how complexes are colored, that for something to be colored it needs to absorb visible light, and the color that we see is a complementary color. So that color corresponds to the differences in energy when the electron gets excited. So there's a larger difference in energy change here versus here, and that's why we see different colors. And the differences in colors allows us to identify what species is that in solution. So I'd like to talk to you about another chemical equilibrium, and that's the binding of oxygen to the hemoglobin in your blood. So the protein hemoglobin, we're going to abbreviate that HB, is found in red blood cells, and it reacts with O2. And this enhances the amount of O2 that can be carried through the bloodstream. So when the hemoglobin binds the oxygen, it allows the oxygen to be transported to where it needs to be in the body. So remember that HB represents the entire protein, and it's not a chemical formula. So we have a picture here. This is the hemoglobin protein. And there are it's a tetramer protein, so there are 4 heme centers. So this is one of the heme centers, 2, 3, 4. And at each of those heme centers you have a porphyrin ring, and an iron sits in the middle of that porphyrin ring. And at that iron center is where the oxygen can actually bind. Now when the hemoglobin binds oxygen, it's very important that this is a reversible reaction, because if it binds the oxygen and carries the oxygen where it needs to go, it needs to be able to release that oxygen. So this is why it's important that it's a dynamic equilibrium. So this is what we mean by this double-headed arrows. Now, even though in the PowerPoint I've got these arrows drawn in my slides, it really should be represented as double-ended arrows like this to show the equilibrium. So it tells us that the reaction is going in the forward direction, but it's also occurring in the reverse direction. So this is what we mean by a dynamic equilibrium. Something's active going on, and it's maintaining that equilibrium. Now, for the hemoglobin equilibrium system, the concentrations of hemoglobin, oxygen, and the oxygen-bound hemoglobin are all interdependent. So they all are interconnected to each other. So if you change one, it's going to change the other. The relative amounts of hemoglobin, oxygen, and oxygen-bound hemoglobin at equilibrium are related to a constant called the equilibrium constant, and we're going to abbreviate that as capital K. So the larger the value of K, the more product is found at equilibrium. Changing the concentration of any one of these necessitates changing the other concentrations to reestablish equilibrium. This is because they're all interrelated to each other. So you change one, the other concentrations are going to change as well. So again, we want to look at that O2 transport. The hemoglobin, we said, had a heme center with an iron. So here we have the iron, and it's actually sitting below the plane of this ring. And there is a histidine below the iron that helps to bind the iron center, and another histidine above. When the hemoglobin is exposed to oxygen, the O2 binds, and it binds in this bent fashion. And the nearby histidine helps to stabilize the binding of the oxygen to the iron. And one of the things you may not be able to see from this figure is that when the oxygen binds, it actually causes the radius of the iron to contract. And as the iron gets smaller, it actually fits within the ring better, and so it goes and it's nearly planar with the ring. So here we have the reaction of our equilibrium. The hemoglobin plus the oxygen goes to make the oxygen-bound hemoglobin. Now in the lungs, with high concentrations of oxygen, the equilibrium shifts to combine the hemoglobin and oxygen to make more of the oxygen-bound hemoglobin. So when oxygen's high, the equilibrium shifts to make more oxygen-bound hemoglobin. Then when oxygen is low, when you get to the cells, the equilibrium shifts to break down the oxygen-bound hemoglobin to make more HB, the hemoglobin, and oxygen. So this releases the oxygen so it can be used by the cells. So this is how we see how changes in concentration can affect the equilibrium. When a reaction starts, the reactants are consumed and products are made. When we're talking about a reaction and the way it's written, we're going from reactants to products, we say that's the forward reaction. Therefore, the reactant concentrations decrease as we go in the forward direction, and the product concentrations increase. As the reactant concentration decreases, the forward reaction rate decreases. So as you start to lose some of your reactants that you're making product with, the reaction is going to slow down as you consume up some of those reactants. Eventually, products can react to reform some of the reactants. So this is the reverse reaction, where we go from products to reactants. And for the reverse reaction to occur, we have to assume that the products are not allowed to escape. So let's say in the forward reaction, you're evolving off carbon dioxide gas. If you do not trap it into the system, you don't have a closed system that CO2 can escape. And if the CO2 escapes, you don't have product to reform the reactants to go in the reverse direction. So you need to make sure that everything stays within the system. Nothing is allowed to escape or be removed to establish an equilibrium. And as the product concentration increases, the reverse reaction rate increases. So the more product that you make, the faster the reverse reaction is going to become. And processes that proceed in both the forward and reverse directions are said to be reversible. This means that you can go from reactants to products and products to reactants. Here we have a hypothetical reaction where we have two red, let's say spheres, going to make blue spheres. And we have some data. So we're looking at time increasing in intervals of 10 seconds. We start off with 0.4 molar red. Remember, anytime you see brackets around a compound, this means concentration and molarity. So we're starting off with 0.4 molar red, but we have no blue, we have no product. And we see that as the red is getting converted to blue, we see this decrease in the red reactants. But eventually this starts to level out, and the concentration isn't changing. We see the same thing happening with the blue, except now we're forming blue. So we start off with 0 blue, and we start making a little bit, we start making some, but the changes get smaller and smaller until there is no more change. Now you're probably looking at this data, and it's a bit overwhelming. So this is why chemists like to graph things. That way you can easily see what's going on rather than trying to interpret all these data points at once. So one of the things that we should notice is that the reaction slows over time, but the red molecules never run out. At some time between 100 and 110 seconds, the concentrations of both the red and blue molecules can no longer change. Thus, equilibrium has been established. Be careful and notice that the equilibrium does not mean that the concentrations are equal. It means that the reaction rates in the forward and reverse are equal. The reaction rates are equal going in both directions, not necessarily the concentrations. Because see, once these start to level out, 0.164 does not equal this 0.118. So it doesn't mean that the concentrations are equal when you reach equilibrium. Once equilibrium is established, the rate of red molecules turning into blue is the same as the rate of blue molecules turning into red. So here's that same information presented in a graphical manner. So we have time on the x-axis in times of seconds. On the y-axis we have concentration. If you will remember, we started off with red and we're making blue, so we started off with a 0.4 concentration. And as time progresses, we start making more and more blue. So we see the decrease of the red and the increase of blue. And if you look at the slopes of the line, notice that the slope is related to the tangent line, so the slope relates to the rate of the reaction. So initially it starts off faster and then eventually it slows until it becomes constant, so you don't have a change anymore. So after this point, we see that the concentrations aren't changing, and this tells us we've reached equilibrium. So let's look at this in a little bit more detail with some reaction dynamics. This time we're looking at time on the x-axis and the rate of the reaction on the y instead of concentration. So initially only the forward reaction takes place. So the forward reaction, we're looking at this blue dot, it's high. And because we don't have any product, the red reaction, so this rate of the reverse reaction, can't occur. So that rate is 0. As the forward reaction proceeds, it makes products and uses reactants. Because the reactant concentration decreases, the forward reaction slows. As the products accumulate, the reverse reaction speeds up. Eventually, the reaction proceeds in the reverse direction as fast as it proceeds in the forward direction. At this time, equilibrium is established. Once equilibrium is established, the forward and reverse reactions proceed at the same rate, so the concentrations of all materials stay constant. Now we're going to look at a more specific example rather than just hypothetical red and blue. We've got hydrogen gas combining with iodine gas, forming two equivalents of HI gas. At time 0, there are only reactants in the mixture, so only the forward reaction can take place. Here we see the H2 is represented by the two white spheres, so this is a diatomic molecule, and the purple spheres represent the diatomic iodine gaseous molecules. And if we were to count the concentration, we have 1, 2, 3, 4, 5, 6, 7, 8 I2s, 8 H2s, and no HI. At time 16, there are both reactants and products in the mixture, so both a forward reaction and reverse reaction can take place. And if we were to count this up, notice that we've now produced 1, 2, 3, 4 of the HI. At time 32, there are now more products than reactants in the mixture. The forward reaction has slowed down as the reactants run out, and the reverse reaction accelerated. So now we can count 1, 2, 3, 4, 5, 6, 7, 8 of the HI. So we've increased the concentration of our products still. At time 48, the amounts of products and reactants in the mixture haven't changed. The forward and reverse reactions are proceeding at the same rate. Thus, the reaction has reached equilibrium. So let's see, we have 1, 2, 3, 4, 5, 6, 7, 8 HI. So we haven't changed the amount of HI in there. So this is just graphical representation of the data that we've looked at, where we have time on the x-axis and concentration on the y-axis. Remember, we're starting off with I2 and H2 as our reactants. We have higher concentrations of those as we start to consume our reactants. We start to lower the concentration, and as we lose our reactants, we're making products, so the concentration of the HI is increasing. And eventually, this starts to level out and the concentration does not change. So at this point, we've reached this dynamic equilibrium. All the concentrations of the reactants and products are not changing. Now the reactions are still proceeding. You've still got reactions going in the forward direction and the reverse direction. It's just you see no net change in the concentrations. So as the reaction proceeds, the H2 and I2 decrease and the HI increases. Since the reactant concentrations are decreasing, the forward reaction rate slows down. And since the product concentration is increasing, the reverse reaction rate speeds up. And again, we've reached the equilibrium at this point where we have the dotted line. Once equilibrium is established, the concentrations no longer change. At equilibrium, the forward reaction rate is the same as the reverse reaction rate. Since the HI at equilibrium is larger than the H2 or I2, we say that the position of the equilibrium favors the products. So this concentration of the product is much greater than that of the reactants. So we're making more product, so it lies towards the product side, so it favors the products. Again, we want to remind you that equilibrium does not mean equal concentrations. The rates of the forward and reverse reactions are equal at equilibrium. That's what's equal. But it does not mean that the concentrations of reactants and products are equal. Some reactions reach equilibrium only after almost all the reactant molecules are consumed. We say the position of equilibrium favors the products. Other reactions reach equilibrium when only a small percentage of reactant molecules are consumed. In this case, we say the position of equilibrium favors the reactants. To describe these equilibrias, we use the equilibrium constant. Even though the concentrations of reactants and products are not equal at equilibrium, there is a relationship between them, and the equilibrium constant relates them. The relationship between the chemical equation and the concentrations of reactants and products is called the law of mass action. For the general equation A plus B goes to form C plus D, the law of mass action gives the relationship below. So K represents the equilibrium constant, and we have the products over the reactants. And you're going to hear this a lot as we go through this semester, especially as we talk about equilibria constants. It's always products over reactants. Here we have the concentration of C, and that's what the brackets mean, the concentration in units of molarity, times the concentration of D, the other product, over the concentrations of the reactants A and B. Notice that if there's a coefficient, so the coefficient for A, we'll have this lowercase a, becomes the exponent on the concentration. And the coefficient for B also becomes the exponent. And again, the coefficient for C becomes the exponent here. And the coefficient for D also becomes the exponent. So be sure that your chemical equation is balanced, because the coefficients in front are very important to the mass action equation. You have to have the balanced chemical equation to write the correct mass action equation. So these lowercase letters represent the coefficients of the balanced chemical equation, and it's always products over reactants when we write the equilibrium constant expression. And this K is called the equilibrium constant. Now, because we're expressing ratios of the molarities or the concentrations units will cancel out. So the K is always unitless. So again, looking at a generic equation for the equilibrium constant expression, where we have A plus B going to C plus D, it's products over reactants, and the coefficients become the exponents. Here we have an example to practice with. We have two equivalents of dinitrogen pentaoxide, and that decomposes into 4 equivalents of NO2 and 1 equivalent of oxygen gas. So if we write the equilibrium constant expression, or the mass action equation, K equals the products over the reactants. So one of the products is NO2, and the coefficient for that balanced chemical equation is 4. That is our exponent for NO2, times the concentration of O2. There is an understood 1 in front of the O2, and that's raised to the 1 power. Again, we're looking at products over reactants. So our reactant is the dinitrogen pentaoxide, and remember it took 2 equivalents of the dinitrogen pentaoxide, so it is raised to the 2nd power. So the equilibrium constant expression is as shown. So what does the value of the equilibrium constant imply? When the value of the equilibrium constant is much greater than 1, we know that when the reaction reaches equilibrium, there will be many more product molecules present than reactant molecules. Because remember, K is products over reactants. So if K is large, we have a larger number in the numerator than the denominator. So this means the position of the equilibrium favors the products when K is much, much greater than 1. And when the value of K equilibrium is much less than 1, we know that when the reaction reaches equilibrium, there will be many more reactant molecules present than product molecules. We say that the position of equilibrium favors reactants. So if K is much less than 1, the reactants are a larger value, so you have a larger number in the denominator than you do the numerator. So the K is much less than 1. The equilibrium favors reactants. So let's look at this in a little bit more detail. We're going to look at the large equilibrium constant first. When the value of K equilibrium is much greater than 1, we know that when the reaction reaches equilibrium, there will be many more product molecules present than reactant molecules. In this case, we're looking at hydrogen gas combining with bromine gas to form HBr gas. So this is a balanced chemical equation. When we write the mass action equation, the equilibrium constant K is equal to products over reactants. Our product is the HBr, and the coefficient is 2, so it's raised to the second power. And our reactants are the H2 concentration times the Br2 concentration. So this would have a large number when it favors the product. So let's see, here we have 1, 2, 3, 4, 5, 6, 7, 8, 9. So K equals 9 squared over, I see 1 hydrogen molecule, 1 times the Br2 concentration. I only see one of those. So in this case it would be 81. So in this case, the reaction lies to the right, and there are much more products than there are reactants, and the K value is very large. Now we're going to look at the opposite of that. When the value of K equilibrium is much less than 1, we know that when the reaction reaches equilibrium, there will be many more reactant molecules present than product molecules. In this reaction example, we're looking at nitrogen gas, N2, combining with oxygen gas. Again, both of these are diatomic gases. They will combine to make 2 equivalents of nitrogen oxide. If we write the mass action equation, the equilibrium constant K equals the products, the NO, raised to the second power over the reactants, so the N2 concentration times the O2 concentration. So if I look at this, I'm trying to count up the NO molecules. I count 1, 2. So K equals 2 squared, and the number of nitrogen molecules, 1, 2, 3, 4, 5, 6. 6 times 4 oxygen molecules. So 4 over 24. So this number is smaller than 1. So this is demonstrating that the K value is less than 1, and we're favoring the reactant side of the equilibrium. So the position of equilibrium favors the reactants. This is the end of our videos for the first part.